When we talk about genomic mechanisms in cardiovascular diseases, what we really mean is how changes in our DNA, from single-letter swaps to big structural differences, affect the heart and blood vessels. It sounds technical, but the ideas behind it are pretty intuitive. Genes make proteins, proteins do the work in cells, and when something in that chain is altered, heart function and blood vessel health can change in predictable and sometimes surprising ways. First, it helps to split things into two broad categories: rare, single-gene, monogenic disorders and common, complex diseases driven by lots of small genetic effects plus the environment. On the monogenic side, you have things like familial hypercholesterolemia, caused by mutations in LDLR, ApoB, or PCSK9, which leads to very high LDL cholesterol and early atherosclerosis. Or hypertrophic cardiomyopathy, often tied to mutations in sarcomere genes like MYH7 or MYBPC3, where a single mutation can drastically alter heart muscle structure and function. Long QT syndrome, arrhythmogenic cardiomyopathies, and some congenital heart defects also fall into this single-gene realm, and they teach us clear cause and effect lessons about how specific proteins and pathways matter. Most cardiovascular disease, though, is polygenic. Coronary artery disease, hypertension, and most heart failure cases are influenced by hundreds or thousands of genetic variants, each nudging risk up or down a little. Genome-wide association studies, GWAS, have been a huge driver here. They've mapped many loci linked to coronary disease, blood lipids, and blood pressure. A famous example is the 9p21 region. It doesn't code for a protein, but variants there clearly increase risk of coronary disease, probably by changing regulation of nearby genes involved in cell cycle control and vascular health. Other notable genes include SORT1, influencing LDL levels, LPA, lipoprotein A, and APO, lipid metabolism and inflammation. Non-coding DNA turns out to be hugely important. Most GWAS hits sit outside protein coding regions, in enhancers, promoters, or other regulatory elements. Changes there can alter when and how much a gene is turned on in specific cell types, say endothelial cells lining a blood vessel or macrophages in an arterial plaque. Chromatin organization and 3D genome folding matter too, because a regulatory element may be far away in linear sequence but loop to touch its target gene in 3D space. Epigenetics gives us another layer. DNA methylation, histone modifications, and chromatin remodeling change gene expression without altering the sequence. Environmental factors like smoking, diet, and stress can alter epigenetic marks, linking lifestyle to gene activity. MicroRNAs and long non-coding RNAs also fine-tune gene expression and are implicated in heart failure, hypertrophy, and vascular inflammation. Mitochondria deserve a shout-out as well. They carry their own DNA, and mitochondrial mutations can impair energy production in heart muscle, sometimes causing cardiomyopathies. Since the heart is an energy-hungry organ, even modest mitochondrial dysfunction can have outsized effects. A newer, surprising player is somatic mutation, mutations that accumulate in cells during life. Clonal hematopoiesis of indeterminate potential (CHIP), where blood stem cells carry mutations in genes like DNMT3A or TET2, raises the risk of coronary disease. The mechanism seems inflammatory. Mutant blood cells are more prone to producing inflammatory signals that accelerate atherosclerosis. How does this knowledge translate into care? In several ways. Genetic testing is now standard for certain cardiomyopathies and familial lipid disorders, allowing family screening and earlier intervention. Polygenic risk scores, PRS, are being explored to stratify risk for common diseases. They're not perfect, but they can identify people who might benefit from earlier lifestyle or medical interventions. On the therapeutic side, understanding genes led directly to drugs. PCSK9 inhibitors lower LDL cholesterol because of the role of PCSK9 in LDL receptor recycling. Antisense oligonucleotides and RNA-based therapies are emerging tools for targeting specific transcripts, and gene editing, CRISPR, is being tested in preclinical models and early human trials for some inherited heart diseases. That said, there are limits and challenges. Many GWAS signals have small effects and uncertain mechanisms. PRS can be less accurate in non-European populations because most studies were done in Europeans. Interpreting variants of uncertain significance in clinical genetics remains a headache. And altering genes or gene expression in humans raises safety and ethical questions. Looking ahead, single-cell sequencing, better functional annotation of the non-coding genome, and more diverse genetic studies will sharpen our picture. Combining genetics with longitudinal clinical data, imaging, and environmental exposure information will help move us from statistical associations to causal mechanisms and, eventually, more precise prevention and treatment. In short, genomics has reshaped how we think about cardiovascular disease. It's not just cholesterol or blood pressure anymore, it's a complex interplay of genes, regulators, cellular states, and lifetime exposures. The more we learn, the better we can identify who's at risk, why they're at risk, and how to intervene in a targeted way.